Welcome to Lab 2, Anatomy of the Cell and Cell Division. This lab will cover pages 6 through 11 in your lab book, and we'll begin on page 6. The information for this lab is also found in chapter 3 of your textbook. So objective number 1 is regarding the plasma membrane, the cytoplasm, and the nucleus. When we look at a typical cell, they have those three things in common. There is a cell membrane or plasma membrane, which is shown in this diagram. From your textbook. The plasma membrane is this white outer covering, but it can be any color. Typically it's transparent, but this is the plasma membrane, the outer covering of the cell. The inside, the fluid substance found inside the cell where the organelles are suspended, is known as the cytoplasm or the cytosol. Either term is acceptable, but if you see a pin or a label just freely inside the cell, not on any particular organelle, then we're looking for you to identify it as the cytoplasm. Cytoplasm gives the cell its shape, and it also serves as a medium for metabolic reactions to occur. The nucleus is this large structure. It's normally centrally located, but not always centrally located in cells. We can identify the nucleus because it has these holes in its outer membrane, which are called pores. That way things can come and go from in and out of the nucleus. And the nucleus is not a solid organelle. It is actually a hollow organelle, and what it contains is DNA. So when we say that the function of the nucleus is that it's the control center for the cell, that just means that it contains DNA, which contains the recipes for basically making any protein that that cell might require to survive. So the nucleus is a space, and inside that space is DNA. We also see a smaller organelle inside the nucleus, which is known as the nucleolus, and that's described under, or that's listed under objective number 3. The role of the nucleolus is to make ribosomes. So that's the function of the nucleolus. Objective number 2 talks about the basic organelles of the cell, and the first one listed here is the rough ER. You can identify the rough ER because what makes it rough is the presence of ribosomes on its surface. The endoplasmic reticulum, or the ER, is identified as being a network of membranes that extend throughout the cell. But the rough ER always begins at the nucleus. We'll see it's continuous with the nucleus in terms of its membrane, and then it extends away from the nucleus throughout the cell. So again, the rough ER has these dots on the surface, which are ribosomes, and the function of the ribosomes is to make proteins, and the rough ER will take those proteins made on its surface, and it'll modify those proteins to make them into their final form. So that's the rough ER. That's the one that's listed second in your objectives. The first type of ER is the smooth ER. And the smooth ER is not shown in this PowerPoint, but it's simply the same as a rough ER, except it does not contain ribosomes on its surface. So here's an example here of the smooth ER. It does not have ribosomes on the surface, so its job does not involve proteins, but it involves carbohydrates and lipid production, or fat production. So that's the smooth ER. Again, a network of membranes that extends throughout the cell, but these do not contain the ribosomes. That's what makes it a smooth ER. So the ribosomes, again, can be either attached to the rough ER, or they might be free-floating in the cytoplasm, as we can see here. The function of the ribosomes are to make proteins for the cell. Examples of proteins might be collagen, which helps give our skin its resiliency. It might be insulin, which helps us transport glucose into our cells. There's a number of different proteins in the body, and ribosomes are responsible for their production. The Golgi apparatus is another network of membranes, but it doesn't extend throughout the cell like we see with the rough and smooth ER. The Golgi apparatus is more a cluster of membranous sacs that are isolated in certain regions of the cell. It almost looks like it's pinched together in the center. The membranes are real close together. And the function of the Golgi apparatus is to take the modified proteins from the rough ER and package them into these vesicles. Here we can see these round structures. Those are secretory vesicles, which are basically little outpouchings of the Golgi apparatus that have been pinched off and contain the proteins made by the Golgi, or I'm sorry, packaged by the Golgi apparatus. So the ribosomes make the proteins, the rough ER modifies the proteins, and then the Golgi packages these proteins to be exported to be used inside the cell or outside the cell. So the Golgi apparatus you'll always see isolated as a network of membranes at different spots throughout the cell. The mitochondria are kidney bean shaped organelles, and they're scattered throughout the cell, and their job is to make ATP, so they provide energy for the cell. We mentioned last time in our notes on the chemistry of life that ATP is the energy molecule for cells. So the job of the mitochondria is to take the glucose and oxygen that we bring in through breathing and through our diet and convert that to ATP energy. So the mitochondria are the powerhouse or the energy manufacturers for the cell. And in organs where we need lots of energy for continuous life and movement, for example, the heart, which we need throughout life, and it's constantly beating, we would expect to see a lot of mitochondria in those cells. The same thing in athletes who are working their muscles for bike riding or swimming or running. Those cells are going to have lots of mitochondria to provide the energy for those activities. The centrioles are special structures that are used for cell division. We're going to talk about mitosis in the second half of this lab, and we'll learn about how the centrioles play a role in creating what's called a spindle that attaches to chromosomes and pulls them apart during cell division. So basically the function of these is for cell division. And they almost look like that Mexican treat churros, which are deep fried and coated in brown sugar, and they're quite tasty. My kids really like those. That's kind of what those look like. Again, they're called centrioles, and they're involved in cell division. Lysosomes are packaged enzymes, for example, digestive enzymes that are used for secretion outside the cell. So they're membrane-bound sacs that might be found anywhere within the cell. Here we can see the arrows pointing to several different lysosomes, and they are just round circular structures within a cell that contain some digestive enzyme. Because to lyse means to break down or digest. So lysosomes are just membrane-bound sacs with digestive enzymes to break down things in and outside of the cell. They're kind of like the cell's janitor. It helps clean things up. Cleans up used organelles and helps digest food when we secrete lysosomes from pancreatic cells, and might fight infection as well. So that leads us to the second part of our lab, which I will call part 2, and that will conclude this video. But before we end, I want to quickly take a look at a diagram that is out on Blackboard of a cell model. This is an animal cell, and you should be able to identify the parts on this cell model by using the key. And then if you look at the next part of the cell picture, there's a side that does not have a key. So I'm going to go to the blank side, and we're going to talk about some of these structures again. Again, this structure here would be the mitochondria. Again, it's kidney bean shaped, scattered. There's several of them scattered throughout the cell. Here we see a cross section of mitochondria. Here we see a whole mitochondria. So some of these organelles have been cut in half so we can see what the inside looks like. Other ones have been left intact. Here is an example of the Golgi apparatus. Here the Golgi apparatus has been cut so we can see the membrane inside. Here are a pair of centrioles involved in cell division. Here we can see a nice smooth ER, no ribosomes attached. And then if we go down a little bit here and look at the nucleus, we should be able to find the rough ER. Here's the rough ER in blue attached to the nucleus, shown in purple. That's the nuclear membrane. And we can see these little dots on the surface. They're kind of orangish pink in color. Those are the ribosomes covering the rough ER. This is the nuclear membrane. This is the nucleus inside the cell. And we see that there's these little hazy squiggly lines here. That is representing DNA, which is in an unorganized form in this model. And that unorganized form of DNA is called chromatin. This is in Objective 3 of your lab packet. Chromatin is uncoiled DNA. Think of chromatin like a plate of spaghetti. Very disorganized strands of DNA found in the nucleus. when that DNA becomes ready to divide into chromosome and forms chromosomes, we see this uncoiled chromatin condensing and forming tightly coiled spring-like structures, which are coiled double helixes or helices. Those are called chromosomes. But when that DNA is very loosely held together, looks like a plate of spaghetti inside the nucleus, we call it chromatin. So if there was a pin asking you what this material was inside the nucleus, you would say chromatin. And again, the nucleolus is shown here, and its job is to make ribosomes. Here we can see some lysosomes. Here's one lysosome, here's another lysosome, here's another lysosome. Just these membrane-bound sacs with digestive enzymes inside. So again, the Golgi apparatus is shown in red, the mitochondria scattered throughout for making energy, the smooth ER, a network of membranes that does not contain ribosomes, and the rough ER that does contain ribosomes, which is attached right against the nucleus. So this purple structure is the membrane of the nucleus. And that concludes part 1 of the cell for Lab 2.